Thanks everybody for being here and for the organizer to give me the opportunity to share my part of my journey with Nextflow with you. And in particular, today I will discuss 6 of the most common challenges that bioinformatics core deal with every day. Here is an example of report generation, which is an important thing. And with Nextflow, I will show you in the presentation today how you can actually achieve a report generation alongside the run of the pipeline that you can share immediately with the users. But let me start with some introduction about Nextflow itself. I don't have to explain to the audience what a workflow is, but I would like to remind a couple of important elements of a workflow. When we define what a workflow is, in the sense of getting from your data to the results of your analysis, usually you define a series of steps where you get your input data in, and then each step produces some outputs, and then you get to define results. Now, if you want to be formal about the definition of the workflow, each of these steps has to be described in a formal way, which also means that you need to describe accurately what kind of data you're going to use as an input, as an output, what kind of software, specifically the version of the software, when you're going to run it, and keep track of all these kind of things. Now, as a vocabulary tool in Nextflow, the definition of one of these steps is called a process. A process definition is actually relatively simple in terms of code structure. You can see here that in this snippet of code, you have the 3 important elements of defining one of these steps or process, which is what kind of inputs the execution wants, what kind of outputs is going to produce, and the code that you're actually executing here. Then, of course, you know that these are not the only things that are usually important when you execute some code. You also need to define what kind of container, if you have a containerized application, where you're going to store your data, and what kind of computational resources you are assigning to this execution. But this can be done in several ways inside of Nextflow, and we will see some of these along this presentation. Now, the key element of Nextflow, from which the name is stemming from as well, are these channels. So imagine channels as pipes through which data packages flow. Now, channels are what feeds a process with the input data that you have defined, and also that collect the output data that the process has produced. Again, as a vocabulary, the execution of one process on a specific data package is called task. So in this example you see here, we have, for example, a sample sheet which defines 3 different samples that have to be analyzed. They flow through this input channel. They go to the process that we have defined. This will trigger the execution of that process 3 times, each for the 3 data packages that flow into the channel. This means that each execution of the process will produce 3 different outputs that are collected inside of an output channel. Now, this is really the core of how Nextflow works, because it's the flow of data through a channel that defines what actually triggers the execution of a process. And that's very important, because by simply connecting processes together in the definition of the workflow, it's the data flow that determines the execution, what goes in parallel, what goes in serial, what process is depending on another process. And it's as simple as that. The moment you have connected processes together through a channel, the flow of data through these pipes that are called channels determines the triggering of a specific execution of that process. And that simplifies the way you define complex workflows and also the way you execute these complex workflows into any computing environment. By doing this, of course, there are a couple of important features that every workflow manager wants to achieve, and I think Nextflow excels a lot in achieving these 5, which I also consider a value in bioinformatics: reproducibility, portability, scalability, accessibility, And then you also want the last one, which normally is not mentioned, but is also cost aware. You can achieve all of these things with features that are implemented in Nextflow. And I'm going to talk about some of these because they are often, let's say, not stressed enough. So reproducibility is something that everybody talks about, but implies a lot of things. Transparency, which means that all the methods you're going to use are detailed, they are accessible, they can be retrieved, they can be found in your execution logs, so you can reconstruct what actually happened. All the input files are traceable, so you can always reconstruct what kind of outputs you have produced, starting from which input files, where they were stored, where they're coming from, what kind of data you have used. And then this is almost obvious, consistency, meaning that the same input always produces the same result, of course. Now, portability and reproducibility are, of course, essential, and containers are critical in ensuring reproducibility. In Nextflow, this is quite easy because here you have another snapshot of a process definition where you can see that quite easily in the code, you can enable the execution of some scripts using either Conda, Singularity, Docker, or any other container engine that you can have in mind, Podman, whatever comes to your mind. And it's relatively easy to do this in the code, as you can see, because you just use either the Conda directive or the container directive, and it's mostly based on either a Docker or a Singularity that can be native or streamed from a Docker container. Now, another important element of reproducibility is also versioning. So keeping track of the versions of the software you have used in that certain execution at that certain time. Now, with Nextflow, you can also quite easily collect the version of the software at the end of a process, and then you can emit that into a channel or a file if you want. And then usually at the end of some of the pipelines that are standardized, you collect all this information and you put them in the report at the end of the report that you generate. So this, as you can see, allows you to keep track of all the versions of all the softwares you have used in as many complex as you want workflow. And then you have a nicely formatted table at the end of the report in your pipeline. So you always know what version you have used to produce that sort of results. Now, Nextflow is continuously evolving, also keeping track of what the Nextflow community asks for and how Nextflow is actually used in the community. So something that is very recent is an evolution of channels and the data type of outputs that you see here. So very recently, these kind of channels have been introduced. They are called topic channels, meaning that when you emit a result from a process, you can define a topic for that channel that is produced, and the pipeline keeps track of all the channels which have been assigned to a certain topic label. Now, if you combine these with another thing that's been recently introduced, which is an eval type of outputs, meaning that instead of being, until now you had either a file output or a value output, so a string, whatever value you might have in mind. An evaluation output instead is that kind of thing that usually is produced as a standard output or standard error. So you can define these as an output here in the process definition, and that allows you, instead of writing the code in the way I showed you in the previous slide, to collect, for example, a software version, to collect through the topic all the output channels that had been assigned that label through your pipeline, and then probably more easy than the code that I showed you in the first slide, simply emit these evaluation of the software version into that channel, collect it, print it into a file, and have it in the same table format as I showed you before. Now, reproducibility cannot go alone. It goes hand in hand also with the concept of portability, because it's very difficult that someone is able to reproduce your workflow by using exactly the same computational environment, the same configurations that you run your pipeline on. So it's very difficult that you can reproduce something exactly in the very same conditions. So portability is also essential to ensure reproducibility. Now here you have an example of a configuration file from Nextflow where you can define what are called execution profiles. So by defining execution profiles, you can launch your pipeline with a specific profile, and this specific profile that you have defined here will be applied to that specific execution. So this allows you very quickly to change a computational infrastructure where you're running a pipeline without changing anything else in your code. Now, from the lines that I have highlighted here, you can quickly spot that it's sufficient to change one line in your profile, which is the executor line, to simply make your pipeline portable from different computational environment. And here you have an example of a HPC, a desktop, or a cloud infrastructure. Just by doing this, your pipeline will be translated into the specifics of the execution environment you're running the pipeline on. So you see that this is very easy. If you do this, instead of doing it as a profile, you do this at a process level, either in the configuration or in the definition of the process, you could then very quickly create hybrid computing solutions. That means that you can execute some processes on the HPC and then some processes that would require additional resources on the cloud. And very seamlessly, when the pipeline is executed, it will span through different type of processes, either on the local HPC, on-premise, or on the cloud infrastructure. So this, as you can see, immediately introduces the other value that I was talking about, which is scalability, because by implementing portability in this way, you can also make your pipeline scalable in a very, very flexible way. Now, from what I introduced until now, you can understand that Nextflow is very flexible as a pipeline in the sense that you can code in any language. We were discussing yesterday at lunch one of the reasons why I adopted Nextflow, and one of my answers was that I think it's sort of a third party compared to other workflow managers. It's based on Groovy, is a domain-specific language, which is sort of neutral compared to many other scripting languages you can use in your pipeline. So this allows you very easily to code in any language and implement your pipeline in any language you want. And then in a very simple way, as I showed you, you can implement all the software dependencies via containers and keep track of the version control. But at the same time, Nextflow is also a runtime that allows you portability very quickly. So it's a portable by design, as I showed you. And this allows you to run your pipeline on any cloud infrastructure or on-premise with any sort of scheduler you might like, including your own laptop if the pipeline is quick enough to be run locally. Now, another of the reasons that I mentioned also yesterday when we were discussing how I adopted Nextflow the first time is the existence of a community of users, which is called NF-Core, that continuously discusses how to implement standards and how to keep best practices in using Nextflow for different bioinformatics applications. Nextflow is a huge community. Currently, it has more than 120 GitHub repositories. It has, in these GitHub repositories, more than 2,000 GitHub contributors. And the Slack organization of NF-Core has more than 8,000 Slack users. But for me, as a personal experience, I don't think that these kind of communities are just numbers. For me, it's about this picture here. Participating to NF-Core hackathons or NF-Core events has been always stimulating. And I've always encouraged, for example, members of my lab to participate because I think they give you a lot. They give you the possibility to exchange, to find like-minded people, discuss best practices in your own applications, and they are extremely, extremely stimulating as an experience. What these Nextflow users community has achieved is impressive in the sense that right now we have actually more than 100 pipelines in the GitHub repositories. And then we have a huge amount of components that can be reused, more than 50 sub-workflows, much more than 1,000 different modules, which are definitions of these processes, like I showed, a standard schema for writing the pipeline, and specifically some tooling. And this is very crucial for people who approach Nextflow for the first time. In terms of tooling, the NF-Core tools that have been released, they allow you to create with a standard template either of those things: pipelines from scratch, sub-workflows, modules, create a schema of parameters for your pipeline, lint your code, and also, more importantly, to fetch all necessary dependencies for your pipeline to be run, for example, if you're using an air-gapped environment for your executions. So considering everything I said until now, why anybody would make the effort of learning something new like Nextflow? In my experience, once you have implemented Nextflow, then your pipelines are easier to maintain. They are definitely easier to port. It's much easier to create reports. And then at the end of the pipeline, of course, is also easier to publish, in the sense that writing materials and methods, keeping track of the versions and everything is a bit of a breeze if you keep your work practices with Nextflow in this way. Of course, you have to learn a new syntax, so you have to make the effort of learning something new. And sometimes you also have to rethink a little bit your workflow with the logic of Nextflow. And that means also optimizing perhaps your workflow, because by rethinking it, you also introduce changes that are usually for the better. But as you might understand, I might be a little bit biased, but I think that the scale here is on the pros side. And I think once you've done these, you're going to only collect advantages. I remember when I moved from London, I moved back to Pavia. And the first thing I've done as soon as I got access to the local HPC was running one of my pipelines on the local HPC. And just by changing a configuration file, the pipeline ran out of the box. And that made me really, really happy, because you know that when you move environment, you don't have to change your job. You simply change your computational infrastructure. Then it's usually a nightmare to port all your code and take advantage of all the work you've done in the previous years. And when that works just out of the box, is sort of a paradise, I would say. Now, let's get to the specifics of challenges that bioinformatics core experience, I would say, every day. There is no specific priority order in the numbers that I put here. And these list of 6 challenges is mostly based on the experience I had before moving to Pavia in leading the bioinformatics core at the Medicine and Healthcare Products Regulatory Agency in London. Now, first challenge. As a bioinformatics core, you serve a wide range of users with a wide range of applications. You need to run analysis for someone who's doing cancer, human genetics, but you also have to run analysis for people who do mass spectrometry, or you have to run analysis for people who do metagenomics. Now, from what I described, it's quite easy to do all of these things with pipelines that are ready to be used, which are curated by the NF-Core community. There is a huge list to choose from. Here, I just put a non-exhaustive list that spans from genomics to metagenomics, immunology, mass spec, and then you have all the other things like imaging, spatial omics, which is quite a hot topic now, and many more. So you can see that without changing anything, you have best practice pipeline for almost all bioinformatics applications you might think of. So it becomes very easy if you use this kind of framework to join the people who develop this pipeline, to discuss if you might have specific needs, and also use immediately available to be used pipeline from the NF-Core community. Now, I've talked about ready-to-use applications, but the other most common thing is that you need to customize something. The pipeline that is the standard pipeline that works for most of the people, it doesn't work for your specific user or your specific customer. Now, this is pretty easy to do based on what I just described, in the sense that you have more than 1,000 modules that are available there, and then you have tooling that allow you to compose your own pipeline using those modules. But if that is not enough, then using the NF-Core tooling, you can also create your pipeline from scratch. This is an example of running a one-liner command with NF-Core tool, where you run NF-Core pipelines create, you give the name of your pipeline, and then in a breeze, you can see here on the VS Code, you will have a whole skeleton for the pipeline with lots of things that have been pre-compiled for you using the standard template that NF-Core has agreed. And you can see there's already a lot of code with placeholders where you have to change some things that highlight what you have to customize. But in really a moment, you've seen that I was able in this video to create a skeleton for any sort of pipeline you might want. Now, pipeline from scratch, but customization also applies, for example, to the core of the portability I discussed, which are containers. Now, for containers as well, you have two options. The easy way, and Sequera has recently released what's called Sequera containers. You log into this platform, and then you can compose a container using the tools that are taken from Bioconda, PIPY, Conda Forge, and then you can, from here, immediately have access to the URL of the container that's built in a breeze, and then you can build it also for Docker, for Singularity, for any platform you might like. Now, if this is too easy for you, you can use also the Pro version of this approach, which uses Wave. Now, Wave is another tool that has been released last year. Waves is a sort of an intermediary between the user and a container registry. It builds container also on the fly, meaning that you can pass a Wave command inside a Docker run command. And Wave is capable to build very, very quickly a container for you, starting from Conda packages, for example. It's the engine behind the Sequera container that I just showed. And it facilitates these kind of operations in the sense that when the user requests for a container, while the image is returned very quickly to the user, the image can also be pushed to a registry. So when the user requests for the image a second time, the image is just fetched through Wave and then returned immediately to the user. So this allows to quickly build the containers on the fly from Conda packages, and then retrieve the images that exist. Now, this is not the only thing that Wave can do. Another important thing is container augmentation, which means adding things to existing container without having to rebuild them. This is possible because, as you know, Dockers are made up with layers. So what Wave does is simply adding an additional layer to the layers of Docker. And you can do this on the fly as well. You can create a folder where you put, for example, a configuration script or another kind of run script. You pass these to Wave, adding that layer to be augmented, and that returns a URL to the Docker run, which allows you to run on the fly an augmented container in the way you like. Reporting. I'm always that go from easy way to pro way. So the easy way here is running multi-QC, which at the end of the pipeline collects the output of many different processes in your pipeline and produces this very nice, nicely formatted HTML report. Pro way is to engineer your reports. So you can create a module that, like this, collects inputs, collects outputs from many different processes, and then it passes them to another process that runs either an R markdown or a Quarto execution that is parameterized. So this way you can basically compose the report in the way you would like most. Monitoring. I'm going to fly quickly here, but simply the concept here is that you don't only want to monitor the execution of your pipeline, so you know that you can run a 200 sample RNA-seq in 3.5 hours, spend 140 euros, which means 0.7 euros per sample. But you can also optimize your pipeline, because if you keep track of the resources that have been used by each processes, now you know which of the processes have used what percentage of the resources you requested, and this allows you to optimize your run. You can also keep track of the environmental CO footprint, which is very important. And also use a trace file to compose any metrics you might like that is outputted from Nextflow. User accessibility. This is very important because when we run our pipeline, sometimes we would like to focus on large or complex project and have the users somehow be able to run with a certain level of autonomy the pipelines that you write. Now, this is very easy because with an NF-Core template, you can access a website like this where the user might use a graphical interface to compose the parameters that are necessary to run the pipeline. So if the user has a very minimal experience, at least with copy-paste and opening a terminal, then at the end of this configuration on a graphical environment, you can copy-paste the command, run it on the terminal, and the user would be able to launch a pipeline that you have written for them. Even more simply, if you have access to Sequera Cloud platform, for example, you can have a whole bunch of features. You can access data sets that have been composed from sample sheets. You can launch your pipeline either on the cloud or local HPC. And as you can see from this example here, everything is on a graphical interface. So this would be really, really usable by people who don't have much of a bioinformatics experience. Last challenge is training. We all know that training is an issue, not only because we all have staff turnover, onboarding of new people that have been recruited, and this is really crucial. There's plenty of training material you can access now. The training website for Nextflow has been completely rebuilt in the recent past year, I would say. So there's plenty of material that is suitable for everybody, from the beginners to the expert to the one who's really, really into Groovy, and now you can find everything. So wrapping up, I think in this presentation, I probably managed to convince you that Nextflow has many benefits in one solution to address the challenges that are typical of bioinformatics cores. You have lots of tooling and training materials, which should make it no excuse to adopt Nextflow if you want to. And most importantly, you have a community that discusses best practices, which you can join. And that's probably the path that is really fun, I would say. So acknowledgements here. I would like to acknowledge Sequera and the Ambassador Program, who funded part of my journey here. My lab funding, who funded the other part of my journey here. The NF-Core community, and then of course my lab members, and in particular these 5 people who are curating NF-Core or Nextflow pipeline or Nextflow training material. And of course the organizer for inviting me here. And here are my contacts, and I'm happy to take any questions.
Nice talk. Disclaimer, I'm as well work with Nextflow, so I'm part of the community, but I'm wondering, like, for new people, I think that is one of the biggest challenges to get to contribute to the pipelines. How long do you think that a person who is a bioinformatician, has programmed in other languages, other workflow managers even, will take to learn Nextflow and for instance, keep maintaining or keep contributing to the current pipelines?
So in this slide, I showed you the people in my lab who work on Nextflow, and I just want to mention a few names. But here there are interns that have been in my lab for their, for example, for their bachelor internship. And in, I would say, a few months, they composed the pipeline, they wrote the Nextflow code, and now this is the lead, the maintainer of this pipeline that you see here. And there's other people who most recently joined my lab, which are, I think, reasonably proficient with Nextflow now, in less than a couple of months. And this, I have to say, it's thanks to the training material that is around there. It's very easy to access information. So not only taking code that is already there and reusing it, but with the training that is available now, you have videos, you have these Gitpod environments where you can really try out the code. I think you can get reasonably proficient in a very short time.
Thank you. It was a great talk. I saw that the resources you use when you deploy your Nextflow in Google Cloud environment, you run it, and it costs only like 100. You in the cloud environment, Google Cloud, it only costs like 140 euro to run the whole entire pipeline.
Yes.
So I'm trying to understand what kind of pipeline it was, how much of the resource, how did you get it to run that low? Because I know we have had instances in my organization where the minimum we have spent to run 75% of the pipeline was close to a couple thousand. That was very cheap. So I'm very curious.
So that example that I showed is running the NF-Core RNA-seq pipeline, so the standard one that is available for everybody. And in terms of options, we used both Star as an aligner and also Salmon as a pseudo-aligner. So we quantified in both ways. So it was actually quite complete. We ran quantification both with Star plus Salmon and with Salmon alone as a pseudo-aligner on 200 samples. Then the other secret is, of course, using spot instances. You save a lot of money if you use spot instances. And one advantage that Nextflow gives you is the fact that if one of the processes fails, it keeps track of it and it can relaunch the failed process. So now this is particularly suitable for the cloud, because if you're using spot instances, that spot instance goes down and it fails because someone requested for that resource on demand, and you need to relaunch the process. But this is done seamlessly. You don't even realize it. So I think the secret for that cost was mostly using spot instances, and the fact that by running Nextflow, you can use spot instances very easily without having to relaunch anything.
Yeah, actually, thank you for the presentation. Brilliant. More than a question, it's like a suggestion. I started using Nextflow like more than a year ago. Pretty cool. My only suggestion here is that, like, I remember 6 months ago, I started to check NF-Core, and I realized that the processes names are all in like capital letters. So my suggestion is that please tell them that this is like really, I don't know how to say it, but as soon as I saw that, I just don't ever use like the template anymore. So like, yeah, just a suggestion, please tell them to change this. Like,
so from this comment, I
like,
take that you're very sensitive to capital letters.
Like there's a like all capital letters for the process names. I've been like programming since, I don't know, 10 years ago, changing languages, but like all capital letters. Please ask them to change this and I will be part of it.
So my response to this is join the NF-Core community and bring this up into one of the conversations.
Sure. Thank you.